Hello, students. We are starting chapter 14, part 1. In this video, we will be covering these three you must knows: how RNA and DNA are similar and different, and how this defines their roles, the differences between replication, transcription, and translation, and the role of DNA and RNA in each process. We'll focus in this video on what is transcription and translation, but I want you to compare these to what you know about replication from chapter 13. And then how eukaryotic cells modify RNA after transcription. So this video will look at gene expression. How do we take genes and build them into proteins in our cells? Chapter 14, Gene Expression from Gene to Protein. In this chapter, we're going to be looking at the DNA that governs our phenotype, our appearance, and not just our physical characteristics, but also smaller pieces like the proteins that make up our bodies. How do genes provide the instructions for building proteins? And just an application of this, here are some donkeys that are living on an island. And on this island, there's a high population of albino donkeys. And the reason for that is because there's a gene for albinism, and that gene is the instructions for building protein. But in this case, if you have this certain gene, it will build proteins that are not able to synthesize pigment. So it basically stops the ability for an organism to build pigment. And because of that, they're not able to create the pigment that gives them their color. And so there's a high population of albino donkeys. And that's a trait that can be passed down from parent to offspring. So the genes that we're made up of determines our phenotype, what we look like. And that's our key question here. How's the phenotype of an organism determined by its genotype? And here's another practical example. There is a piece of DNA in our cells that has the instructions for building the protein hemoglobin. So here's a hemoglobin protein. And it's made up of many subunits. Here's the quaternary structure of our hemoglobin. And this hemoglobin is found in red blood cells. And its function is to carry oxygen. It holds on to oxygen and it carries it through our blood to all the different parts of our body so that our cells can have oxygen. But if the gene for hemoglobin has a mutation or has some change, then this protein will not be built the same way. It will have some sort of change in these subunits in the amino acid sequence, and it will fold into a different quaternary structure. And then its function will change. So it won't be able to hold oxygen as well. And so it won't be able to carry oxygen through the body as well, leading to something called sickle cell anemia or sickle cell trait. And so, as you can see here, a small change in your genotype in your DNA can have large effects on your physical characteristics, as we'll see more in this video. So first, let's start with what is a gene. So scientists have been trying to figure out, okay, we know about DNA, we know that we have physical characteristics. Mendel told us a lot about how these traits are passed down from parent to offspring, but what's the connection between genes and our physical appearance? Well, some scientists thought that for every gene, remember, a gene is just a section of a piece of DNA, so a certain number of bases, nucleotides in DNA. Scientists thought that one gene is the instructions for building one enzyme. And if this was the case, then that would say that all genes code for enzymes. Is this true? No. There is a code for proteins that are not enzymes, so that is not true. One gene makes one protein. Is that true? Well, that would assume that gene only codes for one single protein. But there are many proteins that are composed of several different polypeptides. Polypeptides are chains of amino acids, and they're each coded by a different gene. So there might be many genes for one protein, and each of those genes builds a piece of the protein, a different polypeptide chain that comes together. So that's not what a gene is. Some scientists say, well, one gene makes one polypeptide, but there are many genes that have the code for only RNA. So you have the gene, and then it builds, we'll look at this in a second, a copy of that gene called RNA, but then it will never go on to build a protein. So that can't be true. Or we can say one gene builds one product. But there are many genes that code for more than one product. So you can have the same gene, but it can actually build multiple different products, which is kind of confusing. So none of these are true. So what does that leave us with? What is a gene? What is the gene instructions for? Well, let's take a look at this quote here. It's problematic to define a gene because one gene can code for several different protein products. Some genes only code for RNA. Genes can overlap, and there are more complications. as you can see here, some genes only code for RNA, and they never actually make it to a product. Some genes code for many different polypeptides. So what is a gene? Well, we are going to simplify it today because there are so many different words that we can use, and we're just going to say that DNA is the instructions for building proteins, but as you can see here, it's a little bit more complex than just one gene to one protein. Genes can also build enzymes, genes can build polypeptides that will form into a protein. One gene can form many different products. So keep that in mind as we go through our lesson. Just a bit of nuance to add to what you already know about DNA to RNA to protein, which is our central dogma of biology. This is the main rule that we follow when we're looking at how our genotype connects to our phenotype, or physical characteristics. And an analogy I like to use for this is kind of like being in the kitchen and cooking. Think of your DNA as like your recipe book, and you take it off the counter in the kitchen and you look, and there's hundreds of different recipes for different cakes. Let's say you want to make a cake. So your DNA is the same way. It has many different genes, thousands of genes on one piece of DNA. And now that DNA is wrapped up in a chromosome, but still you have sequences of nucleotides that have instructions for different proteins. Now, when it's time to build a protein, that DNA is going to be copied into a messenger called messenger RNA. Okay, so this is the copy, and it's a molecule. It's very similar to DNA, but a little bit different. And in the kitchen, this is kind of like if you took your cookbook and just took one page and copied down the recipe on a separate sheet of paper. You're not copying down every single recipe. You're just copying down the one you want so that you can move around the kitchen and that paper can get damaged if you need to, and you're not damaging the original recipe. So in the same way, that's the copy of our DNA. It's just one copy of one small gene, not all the DNA, like replication. And then that RNA will go on to build a protein, but as we know, it could build an enzyme, it could build a polypeptide chain, etc. So what are the steps of protein synthesis? How do we build proteins? So let's start with a eukaryotic cell. Eukaryotic cells have membrane-bound organelles. And as you can see here, there's three main steps: transcription, RNA processing, and then translation. We're going to go in detail into all these steps, but here's just an overview. So we start with our DNA, and this is the instructions for building proteins. And the DNA is found in the nucleus. Step one is transcription. Transcribe means to write, and so we're going to write down a copy of our DNA, and we're going to copy it as pre-mRNA or messenger RNA. We'll look at what mRNA looks like later. But this mRNA is then processed and some pieces are cut out. Now you have messenger RNA. This will leave the nucleus and then we will start translation. So we'll take that code, that messenger RNA code, and we'll translate it into a polypeptide, which will fold into a protein. Now this happens on ribosomes, and these ribosomes can be found on the rough ER, or they can be found free-floating in the cell. Now, our eukaryotic cell on the right is the simplified model. We know that there are other membrane-bound organelles that are not included here that are going to help take our protein product and package it so it can be transported out of the cell. Now, the proteins that are built on the ribosomes that are found on the rough endoplasmic reticulum, the rough ER, those are the proteins that will eventually be passed to the Golgi apparatus, which will take those proteins and package them and finally process them and then put them into vesicles, which will then be eventually transported out of the cell so that they can be delivered other places as a product elsewhere. So that could be useful for cell-to-cell communication, or the cell could be producing a hormone that travels far to another part of the body. But whatever that protein is, if it's a product that needs to be sent out, it will be made on the rough ER. Whereas the free-floating ribosomes in the cytoplasm will build proteins that will stay inside the cell for just normal cell functioning. So it might be building an enzyme for cellular respiration or any other protein that the cell might need. Prokaryotes, a little bit more simple. We have transcription and then immediately translation. There's no separation between the two steps. They can happen at the same time. Right after the mRNA is synthesized, the polypeptide chain will be created. Okay, so we just talked about the central dogma. How do we move information from DNA to protein? So DNA to RNA, we call it transcription. RNA to protein, we call it translation. And for simplicity's sake, we're just going to say that genes code for proteins, but we know that there's more nuance there. Now, DNA can also be replicated. So in our last chapter, we learned about replication, which is just where the entire DNA molecule gets a copy made, right? The two strands separate, and then new strands are formed on top, creating two new strands. But if we want to build protein, we're going to do protein synthesis. and we're only going to copy one piece of the DNA. So let's take a look at this. Again, it's kind of like a broad view, and we're going to go into it more detail. So here's our DNA molecule, and notice we have different colors that represent different genes. So each gene will build something different. So let's look at gene 3. If you were to zoom in at the double helix here, you would find 2 strands of DNA. And you can see the top one here is called our template strand. And notice that it's made up of some nucleotides here. And it's attached to the other side, the non-template strand, or the coding strand, we call it. Now, it's important for you to know the names of these, the template strand versus the non-coding strand. Then we will take our DNA, we will transcribe it, so we'll make a copy. And notice our messenger RNA is down here, running 5' to 3', and we see these bases here. But notice that there's a difference in the base letters here. So instead of T, we have a U, and we'll learn more about that in a moment. And then we're going to translate the code into a protein. And the way we do it is we read 3 of these bases at a time. 3 bases is called a codon, and each codon will pair up with 1 amino acid. There are 20 different amino acids, so depending on what the code is, a different amino acid will come and pair up here. We'll learn more about that later. And then the next 3 bases, our next codon will code with a different amino acid, etc., until you've translated the entire mRNA sequence. So this is the general overview. So just kind of looking up close, something to notice is the directionality here. So our template strand is running 3' to 5', and that's going to be important later. Okay, so something to know about our RNA. So remember, our RNA is a copy of the DNA, but it's a little bit different than DNA. It's still a nucleic acid. It's still made up of nucleotides, but it looks a little bit different. So let's zoom into it. First of all, remember, DNA is made up of nucleotides, a sugar, a phosphate, and a base. And those are monomers. And then if you make a long string of them, as we see down here, here's a nucleotide, 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 connected by covalent bonds here. And then you have another strand on the right-hand side, nucleotides bonded together covalently. And then you have 2 strands bonded through hydrogen bonds at the middle of these bases here. Now, DNA, we have bases that are purines, A and G, so they have 2 rings, versus pyrimidines that are T and C, 1 ring. So that's just kind of some review. But our RNA is a little bit different. So the first is that instead of a deoxyribose sugar, we have a ribose sugar. So the difference is deoxy means no oxygen. And if you look here at this second carbon right here, there's a missing oxygen compared to over here. You have 1, 2. Our second carbon has a hydroxyl group, or OH. So there's an oxygen here. If we look at our nitrogen bases, we notice that there's uracil instead of thymine. So U is going to pair with A, and G is going to pair with C when we're adding our bases complementary to our DNA. And notice that U is also a pyrimidine, so it's the same shape as a T. It's only 1 ring, but just know that it is in the place of T. RNA is always single-stranded, so if you see here, here's a single strand compared to DNA, which is double-stranded. And there's a bunch of different types of RNA. So there's messenger RNA, ribosomal RNA, transfer RNA, or siRNA as well. And these have different functions in a cell. So once again, DNA, we are going to transcribe the DNA and make a copy, and the copy looks like this, RNA. All right, so let's get into the details of how we start protein synthesis. And we're going to start right now with transcription. So we're just going to focus on this part. How do we start with DNA and get RNA? So let's begin here. So transcription. Remember, we're taking our DNA, we're trying to get to RNA. And just to kind of go back for a second, this is happening in the nucleus of a eukaryote, but in a prokaryote, it's happening in the cytoplasm. So we're going from here to this RNA molecule here. So it's going to be important that you write this down and draw these diagrams so you can remember all these terms. So here's our DNA, and we are going to see here a promoter. A promoter is a sequence of DNA that the RNA polymerase, this is an enzyme that's going to do the job of transcribing, is going to bind to the promoter. So that's the part that's telling the RNA polymerase where to start. Because remember, the DNA sequence is very, very long, but we don't need to transcribe the entire thing, just the piece that we want, just our gene. We call that our transcription unit. So that's the part that we want to transcribe. So that's our start point. We call this initiation, where the RNA polymerase will bind to the promoter. Now, the RNA polymerase will unwind the DNA here, and the DNA will separate. And now we have two different strands. So the one on top, we're going to call it the non-template strand. It's running 5 to 3 prime. Our template strand on the bottom is running 3 prime to 5 prime. And our RNA polymerase is bound. And then we are going to start transcribing in red here on the promoter sequence. And we'll continue on here. Okay, so that's initiation. Now we call this the template strand because it's the piece that we are transcribing. This one's just left alone on top. Okay, now notice that the template strand is running 3 prime to 5 prime. Okay, and then we'll notice here what direction is our RNA being formed. Okay, our ribonucleic acid, our single strand. So notice as the RNA polymerase will continue on in this direction, you will see it continuing in this direction, this red arrow. The DNA will become rewound, but we are going to be elongating our RNA transcript. This step is called elongation. So the RNA polymerase will run along in this direction, the DNA, and as it runs, it will unzip. And as it unzips, it will transcribe the DNA. Now, it does it in a 5 prime to 3 prime direction. So this side of our RNA is 5 prime, this side is 3 prime. So we're going to add bases to the 3 prime to continue on in this direction. But we're running, if we look at the direction of our molecule, it's 5 prime on the left to 3 prime on the right. And we know that because we're looking at the direction of transcription. So we can say downstream, or what's coming next basically, is the next part of the transcription unit. But just know that this RNA transcript is being formed 5 prime to 3 prime, but new bases are being formed on this end. Okay, and it's being read from 3 prime to 5 prime. So you start on the 3 prime end and you're making your way down towards the 5 prime end on the template strand. And then finally, we will have termination. So we will complete the RNA transcript and we have our 5 prime to 3 prime RNA molecule, and then the RNA polymerase will detach. And now we're going to go on to the next step. So just to kind of get into this a little bit more detailed and kind of re-explain what I just shared, just so you understand it best. Step 1 is initiation. RNA polymerase binds to the promoter sequence on the DNA. What is the role of the promoter? Well, it tells the RNA polymerase where to start reading, the starting point, which strand to read. So it tells you, okay, the bottom strand and the top strand, that will be important. And then it will make sure that we're going in the right direction. So the RNA polymerase reads the original DNA molecule from 3 prime to 5 prime. So this bottom strand, our 3 prime to 5 prime, we will know what direction the RNA polymerase will go because it will be read from this direction to this direction. So going to the right. Okay. So you can see down here, our RNA polymerase is unwinding, and then we're going to start our RNA transcript. Okay. Okay, so just to zoom in here a little bit more, if we were to look at our DNA sequence, our promoter sequence tends to have this sequence for every gene, and we call it the TATA box. Okay, so on the top here, 5 prime to 3 prime, if we were to read it, TATA, AAA, or if we were to read the bottom one from 5 prime to 3 prime, TTTT, ATA, so the TATA box. And that is going to basically be the sequence where the RNA polymerase can recognize, okay, if I see this sequence, I know that that's where I'm going to start binding and starting my transcript of RNA. So the transcription factors, these are other proteins and enzymes, will bind to the TATA box here. They'll bind to the DNA on the promoter. Remember, the promoter is the sequence. And then that will basically tell the RNA polymerase, and specifically we call it RNA polymerase 2, to bind to the promoter and start the RNA transcript. And we call this the transcription initiation complex, but just know that RNA polymerase is the important piece. Okay, now elongation. The RNA polymerase unwinds the DNA 20 bases at a time. The rest of it is rewound. So only 20 bases are exposed. Again, it's going to read the original DNA from 3 prime to 5 prime, but it builds the RNA from 5 to 3. And this is really important. We always build from 5 to 3. Now, there's no proofreading like there's proofreading and replication because there's not many errors. We're also making many, many copies of the mRNA. The mRNA has a very short life. It's going to be used, and then it's going to be basically degraded very quickly after we build our protein, so it's not worth it to proofread. If there is some sort of mutation or error, only one protein is going to be affected. It's not going to affect the entire body. So there's no proofreading here. Okay, really zoomed in just to kind of make sure everything's clear. Here is our, right now we're in the elongation section of transcription. And notice here our DNA double helix has been unwound. So here, 3', if you follow it down to 5', and that is our template strand. And then here is our non-template strand of DNA. Notice that our RNA in red is being built from 5' to 3'. So we are adding in new nucleotides on the 3' end, and we're going in this direction. So we're moving from 5 to 3. That's really important that you understand that. Now, what's coming in here are individual nucleotides. So the cell, the nucleus, actually has these nucleotides just floating around, and they're going to come in. The RNA polymerase will bring them in and have them hydrogen bond here to the original template strand of DNA. And now you're building your single-stranded RNA. Okay, finally, termination. So RNA polymerase will stop at a termination sequence. So there'll be a part of the DNA that says, hey, stop transcribing. And then the mRNA will eventually leave the nucleus through pores of the nucleus. And usually at the end, the RNA will actually turn into this thing called an RNA GC hairpin. Not super important for you to know, but just kind of interesting. This is what, at the very end of this mRNA, will kind of look like this. And it will basically stop the RNA polymerase from continuing transcription. And it will end the sequence. Okay, so just to kind of review, so we start in the nucleus and we're trying to get our information to the cytoplasm. Genes, where are they? They're on the chromosomes in the nucleus, so your DNA is wrapped up into chromosomes, and genes are individual sections of that DNA. Where are the proteins synthesized? Well, we're actually going to make or synthesize our proteins not in the nucleus, but in the cytoplasm, and we're going to do it with some ribosomes. But how do we get the information from the nucleus all the way to the cytoplasm? We don't take the DNA out. We use a messenger called messenger RNA. The DNA needs to stay in the nucleus so it's not damaged. There are enzymes in the cytoplasm. The cytoplasm is a dangerous place. And so if the DNA is exposed to that, it could be damaged, and then the original code is all ruined. And so that's why you only let the messenger RNA leave. If it's ruined, it's not a big deal. You have your original copy in the nucleus. Just to review some vocab here, this strand that is transcribed, we call our template strand. The non-template strand has another word. We also call it untranscribed or coding strand. That is the original strand. And you'll notice that, just kind of like a little hack here, the mRNA will look very similar to the coding strand. That's why it's called coding. Because if you see here, G, G, C, C, A. The only difference is instead of T's, you have U's. So U, T, C, C, G, G. So the coding strand looks very similar, except T's are U's. So that's just kind of something to notice. Synthesis of the complementary RNA strand happens in this piece here called the transcription bubble. So these are those 20 bases that are exposed by the RNA polymerase. And enzyme that facilitates this, RNA polymerase. All right, now the final step is we need to process the mRNA because now we have our mRNA, but we need to take out some pieces. So that first piece that gets made, we call it a primary transcript. So for prokaryotes, they use that mRNA to build protein, but for eukaryotes, we need some work. The reason for this is we need to protect the mRNA. The cytoplasm, again, is a dangerous place. There's enzymes that threaten it. And there are these enzymes called RNA ACE enzymes that could potentially attack this molecule and ruin it. So we're going to add a cap on the 5' end and a tail on the 3' end. And we'll look at that more in a second. We call the cap the 5' G cap and the tail the poly A tail. Now, our whole goal of editing this mRNA is to edit out these things called introns. And introns are non-coding sequences. So they don't code for the part of the protein that you're trying to build. They're kind of excess mRNA. And recently, scientists have been trying to figure out what these introns are coded for. They're still doing a lot of research, and it's not super clear, but scientists are starting to think that maybe these introns have something to do with gene expression. And what that means is like which genes are turned on, which genes are turned off. And there's a lot more research being done currently. But for now, what you need to know is that introns are cut out. Now, the stuff that's left behind, the coding things, the expressed sequence, the part that's turning into a protein, we call exons. So here is our primary sequence. We are going to add a cap and a tail, and then we're going to chop out the introns, and now you're left with your mature mRNA transcript. We call this process splicing. So when we're splicing, we're chopping off the introns and putting together the exons. All right, how do we protect our RNA? Well, first we need to add a 5' methylated G cap, we call it. Another word is G trinucleoside. So what it is, it's a guanine with 3 phosphates here. This will protect the mRNA from RNase, which is a hydrolytic enzyme. Hydrolytic means hydrolysis. It's threatening to cut these bonds of the mRNA. So that's on the 5' end. And then you'll notice here we have a small region on the mRNA called the untranslated region. And then we have our start codon, and then we have our stop codon. So this is the start. This is the part that includes the nucleotides that we're going to use to build our protein. Then we have polyadenylation signal. This is going to be our untranslated region as well. And then we'll have our poly A tail. So what you really need to know here is that there's a region that codes for proteins. We have a start codon that's going to tell your, later it's going to tell your ribosome, like this is where to start building our protein. And then this is a stop codon saying, okay, stop. This is the extent of all the instructions that we need. And then here's our 5' cap and our poly A tail. So what you need to know from this is what the cap is and what the tail is, and that there are certain regions on the RNA that actually code for the protein. Here is even more in detail. So you can see here are these introns, here are these exons in dark red, and then here's your 5' cap and your poly A tail. Now this is how many bases are in between. And so notice that there's actually only like 146 bases here in this coding segment, and then we chopped off most of the rest of the RNA. We do have this untranslated region. That's just kind of like the section before and after. And then we have our 5' cap and poly A tail, but only this is the coding segment. This is the part that will actually be in the instructions for building a protein. Okay, so we already went over this, but just to review, pre-RNA to mRNA, we're editing out the introns. The actual full word is intervening sequences. That's how you can remember introns. These are intervening, they're in the way, so we need to get them out. We splice together exons, and you can remember that these are the good parts of mRNA because these are the expressed. X means expressed. Expressed means it's going to actually turn into a protein. Now, just some fun facts. A gene is on average 8,000 bases. The pre-RNA will be exact copy, 8,000 bases, but the mature DNA is going to be 1,200 bases. And then that eventually will turn into a protein, which will be at 400 amino acids. So there's a lot of junk that we're going to, we used to think it's junk. Now we're kind of thinking that introns might be important, but just know that they're not actually being used to build a protein. And 90% or more of a gene can be introns. So if you have a section of DNA that's a gene, most of it's going to get edited out anyway. So we need to figure out what that's used for. And then we've already gone through this. Once again, the differences between prokaryote or bacteria and eukaryotes is the time and physical separation. So right here, there's a separation in time between transcription and translation. This is happening at the same time. And then RNA processing, there's no RNA processing in bacteria, but there is in eukaryotes. Let's take a little bit of a close look at splicing. This is when we're editing out the introns. There are molecules called snRNPs or SNRPs. These are small nuclear RNA, and it's a mix of RNA and proteins. Okay, so let's take a look at this image actually. So if this is our pre-RNA transcript, here is a protein in purple and then snRNA, and it's going to make a SNRP. And then it will combine with other proteins to create something called a spliceosome. The spliceosome will find the introns that need to be cut out. They'll recognize them, and they'll basically cut out the introns here. They'll bind and they'll chop it up. They'll hydrolyze those bonds, cut them out. And then they'll paste the two exons together that we need. And then the excised, the cut out intron will be released, and the spliceosome will be released. And now you have your mature mRNA. Now, RNA also can work as an enzyme as well. So as you see here, here is an intron, it's RNA, and it can actually cut itself out and then leave. So it can remove itself, act as its own enzyme, cutting the bonds between nucleotides, between the exon and the intron. And then they will bind together. All right, so that is it for now, and we'll go on to that next one next time.